Hello everyone, greetings to one and all. And this is Sharavardhini. This video is about determinant of antigenicity, which explains about the properties of an antigen. And determinant of antigenicity is a part of immunology. So, what is immunology? Immunology is a study of immune system. Now, let's see the determinants for all type of the antigen. Now, let's see the antigenicity. First of all, we should know what is an antigen. It is a small or a large foreign particle which induces the immunity. And the thing is, all molecules are immunogenic, but not all molecules are antigenic in nature. Now, let's see what is antigenic determinants. This small area of a chemical grouping on antigen molecule that determines specific immune response and reacts specifically with antibody is called an antigenic determinant. The whole antigen does not evoke an immune response. Only a small part of it induces B and T cell response. There are various determinants of antigenicity are involved. There are totally 15 antigenic determinants. They are foreignness, molecular size, chemical nature, physical form, antigen specificity, species specificity, organ specificity, autospecificity and isospecificity, degradability, adjuvants, dose of antigen, route of administration, each factor and genetic factor. Now let's see it one by one. And this picture, the antigen as a part called epitope and the antibody as a part called paratope. Now, the first one is foreignness. For electing an immune response, an antigen must be a foreign substance to the animal, which means non-self antigen. Lymphocyte cells are inactivated that are responsible for recognizing self components during this process. The degree of immunogenicity determines degree of foreignness. Example BSA, bovine serum albumin, which highly immunogenic to rabbit but not for the cow. There are some expectations of some self components like corneal tissue and sperm which are effectively secreted from the immune system. And the next one is its molecular size. Most active immunogens tend to have a molecular mass of 14,000 to 6 lakhs Daltons. Examples are tetanus toxoid, egg albumin, thioglobulin. These components have a high molecular size, which means highly antigenic in nature. Substances like insulin have a less molecular weight, less than the 10,000 Daltons, are either non-antigenic or weakly antigenic in nature. And now its chemical nature. In general, the more complex the substance is chemically, the more immunogenic it will be. Antigens are mainly protein and some are polysaccharides. Protein are more effective than polysaccharides. Some expectations like gelatin, histones and protamines which are non-antigenic due to their low triosine content, which means aromatic radials are low in these compounds. And the next is species specificity. Tissues of all individuals in a particular species possesses specific antigen. The specific antigen antibody reaction in a human blood protein can be differentiated from animal blood protein. And the next is antigenic specificity. Antigen specificity depends on the specific active site on the antigen molecule. Antigenic determinants or epitopes are the region of the antigen which specifically binds with antibody molecule. For protein molecule, it requires 5 to 7 amino acid residues. For nucleotide, it requires 5 nucleotides. Antigenic or multivalent and large antigen possesses hundreds of different determinants groups. And the next is isospecificity. Alloantigens or isoantigens are found to be present in some members in a species, but not in all. They are able to produce isoantibodies or alloantibodies. For the example, human blood is divided into various blood groups on the basis of a human erythrocyte antigens and Rh antigen which are present in the surface of a RBC. And next is organ specificity. Organ specific antigens are confined to a particular organ or tissue. Certain proteins of a brain, kidney, thyroglobulin and lens protein of an eye of one species share specificity with the another species. And the next one is autospecificity. The self-antigens are ordinarily not immunogenic, but in some cases the lens protein of the eye and thyroglobulin and other may act as autoantigens. Some antigens like sequenced antigen are not recognized as self-antigen. And the next is its genetic factor. Some substances are antigenic or immunogenic in one species but not in another. Similarly, some substances are antigenic or immunogenic in one individual but not in others in the same species. And now the age. The antigenicity or immunogenic property of an antigen can also be determined by age. Usually, too young and too old have a diminished ability to elect an immune response against an immunogen. And the next is degradability. Antigens that are easily phagocytosed are more immunogenic in nature. They are broken down into smaller fragments or approximate size which contains the antigenic determinants. Antigens introduced into the body are degraded by the digestive enzymes of the phagocytic cells to be represented by the APC, antigen presenting cells. And the next one is adjuvants. Some substance that can enhance the immune response to an immunogen are called adjuvants. The word comes from the Latin word adjuvare, which means help or aid. The use of adjuvants, however, is often hampered by undesirable side effects such as fever and inflammation. Examples for adjuvants are aluminum hydroxide, aluminum phosphate, and calcium alginate salts. And the next is physical form. In general, particulate antigen are more immunogenic than soluble one. Denatured antigens are more immunogenic than the native form. And the next is the dose of antigen. An antigen can influence its immunogenicity by the way of dose of administration. Dose of antigen above or below which is immune response will not be optimal. And the last one is route of administration. Generally, the subcutaneous route is better than the intravenous or intragastric routes. Antigen administered intravenously is carried first to spleen, whether in subcutaneous moves first to the lymph nodes. And now see what is epitope. Epitope is also called as antigenic determinant. And now let's see what is epitope. Epitope is a part of an antigen that is recognized by immune system, specifically by antibodies, B cells or T cells. Based on this structure and interaction with paratope, the epitopes are divided into two categories, conformational epitope and linear epitope. And paratope is a part of an antibody that binds to the epitope is called the paratope. Now let's see the conformational epitope and linear epitope. Conformational epitope is composed of 3D conformation adopted by the interaction of discontinuous amino acid residues. But linear epitope, it is continuous amino acid residues. Conformational epitope contains 90% of epitopes, but linear epitopes contains 10% of epitopes. And here I gave some glossaries to make you to understand better. And here I gave some web and book references. You may check it out. And that's it, guys. I hope you may understand and it is very useful. Thank you one and all.